Cheers, thank you. We'll do Q&A after both of the presentations because we realize the topic is pretty similar. So we're both talking about training in computational skills. So hold your horses, remember your questions you'll have for me at the end of Todd's presentation. So what I'm going to talk to you about today relates to a paper we published at the end of last year in collaboration with image analysis trainers and image analysts at the Crick and in Cardiff. Where do we come from? So we do teach a lot of courses. These are just a few examples of the ones I've been involved with, or I will be involved with in the coming weeks. And overall, people are pretty happy with our courses. Our feedback is overall very positive, and people recommend these courses to their colleagues. So we thought, well, maybe we are in the good place to maybe write down our experiences so that people that maybe are not feeling confident in teaching image analysis can see our experience and then decide, okay, I can do it. If they can do it, I can do it too. So how is my talk is going to be structured? I'm going to cover a little bit of the logistics of what we think about when we plan a course. Then we'll move on to what happens during the course, and then at the end, what to do after the course. I'm going to keep it short and sweet, hopefully. So first of all, where do we start? Who is our target audience? Who are we holding this course for? Are we targeting students or more like core facility staff, group leaders, a mix of the above? And do we want complete beginners? Or do we want people that already know what they're doing with image analysis? Or do we want seasoned image analysts? Do we want to go the train the trainer kind of solution? And what is the aim of the workshop? What tasks do we want people to learn from us? More importantly, how many people we think we can deal with. We normally teach in a group of instructors. We're normally 3-4 instructors plus, if we're lucky, 1 or 2 helpers in the room. And we think we can have more or less 30 to 40 people in the room. So 1 instructor every 5 students to 10 students, but that's highly depending on the content you want to cover. Okay, before I continue, I realize my slides this time around were very much text heavy, and I hate text heavy slides. So on the right-hand side, from now on, you'll have some entertainment for you. I decided to go with a British artist, David Shringley, very peculiar sense of humor. I really appreciate them. I hope you will too. But the right-hand side of the slide has no like scientific content. It's just for you to kind of don't feel too bored of hearing me talking. Okay, selection process. Our courses tend to be oversubscribed. There are a lot of people that want to learn image analysis and cannot do so easily. Therefore, we had to implement some sort of selection process where people tell us a bit more about what their job title, institution, field of work is, what is their modality of interest when it comes to microscopy, if they already have data, and if they have any experience coding. We also want them to write for us a brief statement. Nowadays, this is mostly ChatGPT, unfortunately, but still we want them to try and justify why they want to be in the course. Personally, between our team of instructors, we tend to prioritize people that will stay longer in research. So we will prioritize core facility staff over PhD students normally, but this is our decision, and it might depend on the setting where you end up teaching. We also like to have people that already have data so that they can kind of go home and start doing something with what they learn. And we normally teach introductory courses, so we want a mix of skills in the room, but mostly beginners. And we try to have a good geographical spread so not to end up with a room full of Londoners. When it comes to software, we advise people to stick to what they know and to make it open source if possible. We do love open source. Make sure you are very strict with yourself on what you want to cover because it's going to take longer than you expect, about 5 times longer than what you expect. We like people to bring their own laptops so that we can sort all of the technical issues there and then, so they can go home and they're ready to roll. They don't have to do installations, and I'll touch upon installation in a bit. And we don't like people bringing their own data. I know this is controversial, but we do prefer to use open data, like data from the IDR or for the bioimage archive, because it makes our life easier. And also within the time scale that we normally have for these workshops, 2-3 days, it's very challenging to kind of meet expectations of people bringing their own data. We want to give them the tools to hopefully be able to go home and start working on their own data, but we tend to avoid letting them bring them on. Quickly on logistics, these workshops tend to cost some money, even just for coffee required to produce code. Per catering, room booking, whatever. But there are funders out there that are happy to help you cover these kind of costs. Here up in the slide, just a couple that have helped us over the years. In terms of room, make sure there's enough space for your participants to kind of sit around the table and interact, and there's enough space for you to hover around. It's very hard to kind of look over people's shoulder if you don't have space without elbowing them. And make sure that the length of the workshop kind of makes sense. You want to it's a fine balance between how much time people can take away from the lab work and how much they want to learn. So we find that 2-3 days is optimal for us, but it might depend on your crowd. And finally, consider a mix of theoretical and practical so that you can check in often on what concept you explain, so you make sure you don't lose people along the way. In terms of material, we start with an overall introduction about theoretical concepts, and then we move on to perform the same exact pipeline on the same exact images, starting with Fiji GUI, then Fiji macro writing, then Python, Jupyter notebooks, and finally Napari. All of this material is openly available. We try to make it as fair as possible. It's on Zenodo. You can scan the QR code or come and find me at some point later, or just Google our names. But we want people to be able to reuse it as much as possible, especially if they need a starting point if they never thought before. During the workshop, a lot of the time will be dealing with technical issues. We have a dedicated session for installations, especially for Python reasons. I've installed Napari on about 200 machines. I still see errors I've never seen before. It's all good. Just dedicate some time for it and make sure that people leave the room with everything installed and working. Manage expectation. There's always be somebody in the room that will expect to leave the room being an expert. Luckily for us, image analysts out there, this is not the case. But remind them that this is just the beginning of a big adventure in the image analysis world. Promote interaction. These workshops are very, very good networking opportunities on top of being opportunities to learn something new. And finally, check in often, have clear milestones, exercises, so that you can make sure that nobody's lost along the way and people leave with something in their baggage. Anyway, after the workshop, get feedback while they're in the room. Don't let them leave before they've completed the feedback form. Otherwise, they will not do it, or at least not all of them. Encourage further learning by giving them different resources for fora, I guess is a plural, publication, blogs, anything that you think can help them in their adventure with image analysis. Publish the content like we do to make it fair, but also to make it easier for the next people to teach. And finally, hopefully like I've been doing today here, share your experience so that other people feel more comfortable. If somebody with bananas on their slide can teach their courses, surely so can you. Okay, with this I'd like to close. Hold your questions and I'll pass over to Todd and then we'll do Q&A together. Thank you.
I just need my talk up. There we go. I could have done Stefania's slides again, but I thought that would have been a little redundant. I'm going to talk a little bit about a course that I teach at the Crick. I'm Todd Fallesen. I'm an image analyst at the Crick. And I teach a course on demystifying HPC. So our beginnings for why we decided to teach this course, this is not something we offer externally just because HPC is sort of, unique to every computing system. Every institute has their own system. But we are looking to try to build this out so that people can replicate what we do. So our beginnings came from, hi, HPC team. I've just obtained visiting access to the Crick, and I was wondering what you could do to help me set up. And then they said, you can do this. And they said, hey, what's an SSH key? And we're just like, oh, okay, so we're gonna go with that. Which a lot of people don't know what an SSH key is, and that's completely fine. So then the next person asked, all right, hi, here's my SSH key, followed from our HPC team saying, you gave us your private key. Can you please send us your public key and delete what you just put on the institute-wide Slack? Yeah, and then I thought that I was being all good. I've written lots of documentation, have our own image analysis wiki to help people get started using HPC for image analysis. And I sent these detailed instructions on how to load Fiji on our on-demand system. And I don't know what on-demand is, LOL. So we decided that we needed to start doing an HPC course. And for everybody here who doesn't know what on-demand is, because I'm going to mention it a few times, is there a laser here? It's green. This is our on-demand desktop that we have at the Crick.
Little hard to hear up here because you're not.
Because I'm nowhere in front of a microphone. I'm sorry. Usually I'm just really loud. So, so here is our HPC desktop, and we can type Fiji on it. We can run Jupyter Notebooks on it, other things. We have Elastic on our HPC system. And it's just all these tools that we let people use. So we decided to start a class on using HPC. And the class itself is image segmentation using machine learning on-demand. And so this is the generic text that they sign up for when they sign up for it. And the whole idea is that they learn how to use our HPC system as like a project-based course. They have a reason to take it other than just sitting in front of it and learning what the command line does, though they have to learn what the command line does. So our motivations for it is we have a lot of huge data sets at the Crick. We've got 3 different light sheet microscopes, lots of confocal. We've got storm data, we've got HRM data. All these things are becoming rather large, or people that just leave the microscope running for a weekend, and they've got huge confocal data sets. And we need to use HPC just to do the analysis on these things. And there's 3 image analysts at the Crick, and we don't have enough time to do all of them. Also the rise in machine learning. So people basically want to use Stardust or CellPose to do everything. Like if they got 3 cells on a slide, they want to use Stardust to segment them. And I'm like, thresholding works, guys. But and lack of training from HPC. And this is probably something that a lot of us have seen throughout all of our different institutes, is that our HPC team themselves, which are nice guys, all guys, but they're nice guys. Yeah, they have never taught anybody how to use HPC before we started doing this course together. So the goal here is to demystify HPC while also getting the HPC team to understand the unique needs of wet lab biologists. Now, that's not saying they do deal a lot with bioinformaticians, and they don't know the difference between a bioinformatician and a wet lab scientist. And so we'll just leave that. So the other reason I started teaching this class is I felt like I didn't want to keep answering the same questions over and over again, because there is just an auto-generated thing I can cut and paste from Slack on how to, install Fiji. And then these are why the students end up taking the class. And you can see, let's see, it's about 25% of them really just took an 8-hour course on HPC and machine learning just to start learning how to use HPC. And then they went off and used it for other stuff, like using RStudio and whatnot. So they weren't just about the machine learning, but thankfully a lot of them did learn about machine learning because I'm image analyst. So like I said, it's a project-based course. I start them off just with this and they get scared, just the command line. And then I teach them about Anaconda. They go through Anaconda, they have to install Stardust. I don't teach them how to use CellPose. It's very similar to Stardust, but it takes much longer. And so when you're in a one afternoon of trying to teach segmentation, it's easier to teach the one that runs quickly. So then they install Stardust, they make it work with GPUs on our cluster in a Jupyter Notebook, which is no small task. And then they start learning a little bit about just running Jupyter Notebooks. Now, they're not doing a ton of hardcore coding because they don't need to do a ton of hardcore coding, but they do need to know how to access their files. They need to know how to troubleshoot if something comes up, who to ask, if it's me or if it's the HPC team. And so they sort of learn these things in the project-based course. And finally, I make them do annotations on their own, and we do train a model. So this is the training of a model that we did over our coffee break. It was not amazing. I had 10 students the last time I did this. They each annotated one slide. We threw it together and trained this for 2,000 epochs over coffee. So you can imagine it was not a very long run. But they managed to see that you can actually train an image, train a model. So they went through the whole thing of then using this then to predict on their images that we saved out. So here's the actual curriculum, if you will, for the morning. So we do this as basically 8 hours. They take an hour for brunch for lunch in the middle. So in the morning, they do learn all the normal HPC things you'd think of: command line commands, file structure, sim linking, because for some god awful reason, we only give them 5 gigabytes of data space, so they have to map to someplace else. Khan environments and Jupyter notebooks, et cetera, all the way through. This is important. GPU versus CPU computing, because a lot of them don't understand the difference, which is fair. And we have to teach them about this so that they can properly do their analysis, because they're trying to do machine learning on the CPU. It's going to take ages. And then they do learn about machine learning basics. So they are not going to come out of this being like TensorFlow experts, but they are going to come out of this understanding a little bit about hyperparameters, a little bit about what they should start changing. So if they have a stardust or a cell pose model, they can train their model, they can change a little bit, like, all right, I need to change the diameters, or maybe the learning rate or something like that. They do learn the importance of the train test split. I make sure they understand this, that it's basically their controls. If they don't do these things, it doesn't work right. And then the accuracy measurements or metrics. And this is sort of our results. You can see, and I was amazed also that I got 20 responsives from about 30 students, so I threatened to beat them. How comfortable you're using the command line before and after. You can see that a lot of them go quite well towards this. This is the bad, this is the good. So we get a broad sweep. People are more comfortable using the command line, and it's true, I do see them often enough that they are able to manage their data pretty well in the command line. And then how comfortable you're using machine learning, we get a pretty good response for that too. And I do see this play out too in the actual users, that they'll be using their Stardust and their cell pose models because they'll ask me about it, because they'll still have questions, but at least I'm not answering the same basic questions 19 times about how do I launch a Jupyter notebook. It's more like, this is giving me weird results and slight pixels off here. Why is this? And I'm like, because your data's bad. But yeah. Anyway, so that being said, we are trying to bring this out further and see what more can be done, because we are not the only game in town. April 28th, if anybody here is based in London, so George and Stefania, we are doing a workshop for bringing together the HPC teams from King's, UCL, Imperial, and the Crick, as well as wet lab scientists and image analysts. We got some funding to bring them together to come up with a curriculum. So the idea is that in the first half of the day, we're going to have wet lab scientists and HPC people give talks to each other so they can see what each other do. Then we're going to give them some beer and pizza, and then we're going to sit them down in breakout rooms, and they're all going to work as small groups of 8 to 10 on what they would put into a curriculum to teach wet lab scientists on how to use HPC. So hopefully by June or July, hopefully by early June / Elmi time, we'll have some results from that we can share with people to see what a good curriculum would be, because I'm sure that mine's not the only one. I'd like to thank the people on here. This QR code links to our materials that we do teach from. So we have a lot of it, of course, is Crick dependent. And our people that helped me design this course, Chris and John, Lewis and Oscar, are the guys at HPC. And Vanessa was our very talented undergraduate student who also taught this course the first time to a bunch of postdocs, which went over really well. So when you're 19-year-old undergraduates teaching the postdocs. So I'd like to thank you.